All right. Good morning, everyone. Thank you for joining us today. Pleasure presenting for you. My name is Claire Sengupta. I am the Field Application Scientist for the Mid-Atlantic region here at VisGen. And today we're going to be discussing highly multiplexed, high-resolution in situ assays using the Merscope platform. We will have ample time at the end for questions where we will be able to hopefully unmute people if they have any discussion that we'd like to foster at the end of the presentation. We also have the Q&A box open during the presentation, and it will be moderated by my colleagues here, Jose, my sales partner, and our colleagues from marketing, Stephanie and Andrea as well. So if you have any questions during the presentation, please feel free to put them in the Q&A. Otherwise, we look forward to discussion at the end of the presentation. And with that, we will get started. And if you choose to, you may also scan this attendance log here. Please do not feel obligated to. You have already registered for the webinar, and we appreciate your attendance and participation. So wanted to start off with something very impactful to see, which is the end result of the Merfish protocol and what you can achieve using the Merscope instrument. This is our visualizer software where you can upload your final results from your Merfish run and see them in total. On the left, you can see the genes in the panel described. This is a mouse brain coronal slice. These are clustered cells here. You can see the populations clustered in the scatter plot on the right-hand side of the software. These are all capabilities of our visualizer software, which is an excellent platform for looking at your Merscope Merfish data. It is free and it is available for download on both Mac and Windows. And you can see here, as this person is scrolling through the data, you can see the true subcellular resolution and the segmentation capabilities of the software and of the data, as well as the high resolution achieved by the Merscope. So just to give you an idea of the power of this technology right off the bat. So how do we get here to the Merfish and protocol and Merscope instrument? So essentially, we are aware as biologists, as researchers, that biological systems are intricately spatially organized. We lose some of that capability with traditional bulk and single cell sequencing. It can show us parts of the picture, but not the total picture because you lose that ability to make spatial associations. With spatial genomics, you can achieve this true single cell resolution as we saw in the previous slide, and it's transformed our understanding of these cell type and cell specific relationships due to this added valuable insight. We can see the bigger picture by doing this highly multiplex direct in situ detection in an intact piece of tissue slice. So specifically, our Merscope platform is a very comprehensive solution to achieve this spatial genomics technology. Right now, we have the ability to run 500 genes in a single run from a custom design panel with whatever genes the end user wants to run on their panel. Very shortly, we will have the ability to support both with reagents and on our platform, 1,000 gene panel in a single run. And this is coupled with the extremely high sensitivity and accuracy that is imbued by the Merfish protocol, which is extremely error robust and uses direct in situ detection of single transcripts. And this is over a large imaging area of a centimeter squared with high resolution. Again, we saw that in the data that I showed from our visualizer video a couple slides back. And this area is important and the resolution is important because then that allows you to not have to do follow-up sequencing. So we'll show some slides later on that really support this in terms of the reproducibility of Merfish results to traditional sequencing. And the added benefit of keeping tissue intact Not only do you get the benefit of spatial genomics, but you can also do protein co-detection in the very same sample, which adds another dimension to your data. The Merscope platform itself is very user-friendly. You saw their visualization software a couple slides back. The instrument itself is very user-friendly, straightforward, provides on-screen prompting. There's very little tinkering that one needs to do or babysitting of the instrument. And on top of that, we have our web-based gene panel application, which supports the construction of these custom gene panels, and in addition offers a plethora of resources, including, like I had mentioned, download of our visualizer software, as well as the most up-to-date and comprehensive protocols, application notes, and SDS of all of our reagents that we offer. So what is the Merscope? So the Merscope combines our Merfish protocol, which is essentially direct quantification of RNA in situ, so SM-FISH, combined with a proprietary barcoding system, which allows us to expand beyond the spectrum of fluorescence that is traditionally used in SM-FISH and design these custom gene panels of more than 500 targets. And together, this allows us to achieve highly multiplexed RNA detection with single cell resolution and subcellular resolution with high accuracy and consistency due to this error robustness that's built in to the barcoding system. Combining this with the Merscope instrument is the entire Merscope automated platform. What we offer at VizGen is truly an end-to-end solution. So I've already talked a little bit about our custom gene panel design. This is a screenshot of our gene portal. Users can sign up for their account and immediately be able to start designing their panel with their custom gene list. Right now, it is set up to design panels for mouse and human. However, as I'll mention in coming slides, you are not limited to just mouse and human panels. We can design a custom panel for any species or subject that you are, the end user, are able to provide an annotated transcriptome for. So that is a large point of flexibility that we can support. Once the panel is designed, and it is quality control checked by our internal teams and delivered, we are able to move on to sample preparation, for which we also can provide all of the reagents for our Merfish protocol, including tissue slides and reagent packs. Once the sample preparation is done, it is loaded into the Merscope in a flow chamber, as you can see here on the right, with our reagent pack, as I just mentioned, to perform the cycles of imaging that lead to the barcoding system. In our Merscope here, so the instrument on the left is our Merscope, and you can see there's two large boxes here. It actually has a very small footprint, so these can be put directly on a lab bench. There's no need for an Airtable or stabilization plate or any secluded dark room. This can be just out in the open in your regular lab space on a lab bench. And these two boxes here on the left, we have the fluidics and microscope part of the Merscope. So within this box is the fluidics, which does the cyclic staining and elution for our barcoding system, in addition to the high resolution microscope, which does the imaging. And on the right, that smaller box is the controller PC and the touchscreen, which controls the instrument. What is not shown, but also included, is the analysis PC. So there's a very high-powered analysis PC to which the data during imaging is transferred over, saving you time at the end. And this is where segmentation is done of your data and produced. The VZG file, our file format, is produced, and that is what you see on the right here on the visualization software. And I would like to add that the output is not just limited to this file format, which you can readily open up in our visualizer software and right away see your experimental results. You also get all the raw data file formats for your sample. So you get a cell by gene matrix, and you get a transcript list and you get a cell coordinates, all raw data in CSV files, and you also get cell boundaries generated by the segmentation, and as well as all the raw images from your run. So again, another point of flexibility of this machine and our platform is that you are not locked into a proprietary file format. You get all of the raw data to do any downstream analysis that you wish. So the Merfish protocol itself is really unique and what drives this technology. And there's three main stages of the protocol. The first would be hybridization of the probes to your sample. So this is what we use, these in unique encoding probes. We generate tens of thousands of probes per panel against to be complementary to whatever genes are in your panel. At this time, we would also do the staining for the protein co-detection. So again, this is just the hybridization phase. The next phase would be clearing of the sample. So this is, if you're familiar with other modalities of tissue clearing for microscopy, such as light sheet or multiphoton, this works much the same way. Utilizing a gel, we remove unnecessary components of the tissue that could interfere with the measurement and the clarity of the measurement, reducing background. So this propels the instrument to allow it to achieve the high resolution, true high resolution with the microscope in the instrument, and allows us to do single spot detection and quantification. Finally, we have the imaging part of the protocol. So once the sample is prepared, it is loaded onto the machine, and then the imaging commences. So this is where I was talking about the cyclic hybridization and extinguishment of the what we call readout probes to the hybridization probes. And over the course of many cycles, the barcode for each gene is generated. And we'll go into that a little bit more in some subsequent slides. And then the end result, again, on the far right here on the bottom, is the single transcript spots that we can visualize in our end data. So stage one of the protocol, again, is hybridizing these unique encoding probes that are complementary and made against your gene panel, your specific genes in your panel. And you can see a little bit of what the actual construction of these probes is here. This black part here is what binds to your actual gene, and these colored parts are the where the readout probes would target. And these are where the readout probes are bound and imaged during those cyclic cycles of hybridization and extinguishment. So just a little bit more detail about the clearing. Essentially, the sample is embedded in a hydrogel matrix. This is very similar to a lot of other tissue clearing protocols used in other microscopy. And once the tissue is embedded, the sample undergoes clearing and digestion. So anything that is not the target nucleic acids and probes is removed. So now we have this very low background sample that is very conducive to imaging of high resolution imaging and that 100 nanometer resolution that we were talking about earlier. And as for the imaging, every cycle, readout probes are bound to the encoding probes here, and they are imaged with 3 different laser lines generating this barcode. So every time fluorescence is detected, it is registered as a 1 bit. Every time it is not for that round, it is registered as a 0 bit. And as the imaging rounds progress, you can see the barcode is built up, and these are then decoded using your code book for your gene panel to represent unique barcodes that were generated for each of the RNA species in your gene panel, which again, as they're decoded, they're assigned pseudocolored uh, pseudocolor assignments, and each puncta is representative of a single transcript here in that image. And one more time, if anyone has any questions about this, we can again discuss this at the end, but just another little infographic about how this barcode is generated. You can see again, every time there is fluorescence detected, there is a 1-bit registered. Every time there is not, there's a 0-bit registered. And by the end, we have an entire barcode generated for each of these unique genes. And because of this barcode and the ability to add bits onto this barcode easily, it allows us to readily expand these panels. So this is how we can expand from a 500 gene panel to a 1,000 gene panel pretty easily on our instrument. And just to give you another idea of what this data actually looks like once all of this process is completed, on the left here, we have an image of human ovarian cancer. There are 8 example genes pseudocolored here in this larger magnification image, and you can see the added information that not just knowing the identity of the genes, but also their spatial representation adds to this data set. This is a 500 gene panel. 174 million RNA transcripts were detected here. And if I can play this video here, we can actually see a higher magnification image looking at the cellular level of this data, and you're able to see all 500 genes in the panel rather than just the 8 here. So as we zoom in, you can see the density of the transcripts, the ability to resolve them at single cell resolution here, and how valuable it is to be able to see where they're actually spatially located. So as I mentioned, in the gene panel portal, you have the ability to seamlessly design a mouse and human panel. And using this, we have over 40 different tissue types that have either been validated for Merfish in peer-reviewed publications or internally. So you can see the list of different tissue we have here in both mouse and human: brain, spinal cord, intestine, liver, ovary, heart. Some of these are traditionally pretty challenging tissue types for histology, but using the knowledge we have generated with our users and internally, we're able to support protocols for preparing this kind of tissue across the board. And again, you are not limited by the type of preservation that you choose as the customer for this tissue. So you can either have cells in culture, fresh frozen tissue blocks, or you can do FFPE tissue. So you're not limited by the kind of tissue preservation. We have protocols and support and knowledge for all of those different tissue preservation types. So again, what does this technology enable you to do? And what it has enabled our users to do is profile genes across these large tissue areas. I've showed you a coronal section of a mouse brain and human ovarian cancer in previous images. We'll look at more soon. And speaking of the human cancer, you can also examine clinically relevant samples because, again, most samples coming from the clinic have different kinds of preservation, such as FFPE. And again, we are capable with protocols and support of preparing those samples for Merfish and Merscope. And using this technology, you're able to characterize cell function and state, which again, we'll be able to see in some peer-reviewed publications closer to the end of the presentation without the need for follow-up sequencing. This allows you, with the spatial dimension, to look at the intracellular organization of the transcriptome. So again, this high-resolution microscope that we have built in to our Merscope allows you to not only look at the spatial relationship at a larger magnification, but also look at the resolved single transcript spots in where they are situated subcellularly. And this, again, allows our users to generate and map out complex tissues, and complex in the way that they might uncover some nuanced observations about the transcriptome as it relates spatially because of the sensitivity of this technology as well. Some advantages of the technology that we've kind of touched on. You're able to do, like I alluded to in the last slide, this quantitative measurement across this large dynamic range of gene expression. So there's two examples here of how Merfish can recapitulate other forms of either direct quantification of RNA transcripts or bulk sequencing. So on the left here, on the y-axis, we have the copy number per cell of Merfish. On the x-axis is copy number from SM fish. And you can see the extremely high correlation of the two data sets. This is from a Merfish publication from PNAS in 2016, and just goes to show the ability to recapitulate established technologies with the Merfish protocol. And importantly, on the right here, the same comparison was done, but with bulk sequencing. So this is copy number per cell of Merfish again, and then bulk sequencing. And you can see over this large range of 4 orders of magnitude, there is a high and tight correlation between the copy number per cell of Merfish and bulk sequencing. But I also want to draw your attention to the lower left-hand side of this graph. So this is essentially down to 0.01 copy numbers per cell. And what that allows you to do is examine low transcript density genes and provide more nuanced data because we don't have to amplify our signal with the Merfish protocol. You're able to actually directly quantify copy number of your target genes, even down to this low copy number per cell value. And speaking of the sensitivity of this technology, another comparison that we'd like to draw people's attention to is between these two publicly available data sets, the Merfish protocol on the top, and the bottom is an array-based platform. This is a single gene called OPRD1. And on the top, we have the Merfish data set from mouse brain. On the bottom is the data set from mouse brain for this array-based platform. And just looking at the transcript density here. So the comparison that we would like to draw attention to is the ability to resolve single transcripts with the Merfish technology in the Merscope and actually localize them to a cell, and the ability to segment them down the line and assign them per cell. With the array-based platform, there's actually a window put on this data, and the grid space is about 55 microns, so it doesn't allow the resolution of actual single cell. And these transcripts are summed over that area, and the end result of this is that there are 70 times more transcripts per gene detected with Merfish. So this goes hand in hand with that large dynamic range of transcripts that you're able to detect with this technology. You have the ability to be more efficient overall in the number of transcripts detected over your tissue area. So getting into some applications of the technology that really drive home some of the capabilities of the Merscope. This technology enables true single cell atlasing. So what we're looking at here is figures from the 2018 Science publication, wherein 155 genes were evaluated in the preoptic region of the hypothalamus in mouse brain. So here we can see the number of cells characterized here. It's over 450,000 cells, and they're sorted by cell type here. And again, you can see this extremely strong correlation with single cell RNA-seq and the Merfish data across these different cell types of inhibitory and excitatory, which is very important. The spatial analysis, again, the spatial dimension of this data is very important, and you can see the distribution of these different cell types over the area of the hypothalamus here, including some unique insights about the integration of certain cell types that were not thought to be integrated in certain nuclei that were observed to be spatially present with this data set. And then importantly, down in this figure on the bottom right, not only are able to describe these cells functionally, but you're able to assess their activity as well. So what we have here is the assessment of CFOS. So this is an immediate early gene that is activity dependent that has been assessed in this data set. in response to different behaviors such as parenting, aggression, and mating. And with a clustering analysis, these researchers were able to observe certain clusters of these cells, upregulating this gene in response to specific behaviors. So this lends itself to be able to describe not only where these cells are at space, but also what their function is. And this next application we have here demonstrates how different cell types interact with each other. So again, this is also lends itself to not only being a spatial study, but also a functional study. So this is looking at the interaction of macrophages with cancer cells. We're benchmarking it again in this left-hand figure against single cell RNA sequencing data. And you can see the strong correlation between the two data sets. And then the spatial component of the Merfish data allows these researchers to measure the relationship between nearest neighbor cells, like these MES, like glioblastoma cells, and their enrichment of macrophages next to this cell type. And in addition to this, they were able to look at the enrichment of certain supporting cells like oligodendrocytes to these glioblastoma cells as well. And that gave them a unique observation about not only immune cells, but also supporting cells enrichment near these, the crosstalk between these populations in this microenvironment. And again, to highlight the throughput, so we kind of touched on this when we were discussing the ability to resolve 70 times more transcripts than an array-based platform. But again, just looking at the number of RNA species here present in just 200 by 200 microns of tissue as assessed by Merfish and the Merscope, this truly enables you to get the most out of your data with no follow-up sequencing needed. And to drive home the utility of this protein co-staining, we have a example data set from human colon cancer. So on the left here, we have our 3 antibodies that were used to stain this tissue. You can see this is the exact same tissue slice. So pan-CK, smooth muscle actin, and E-cadherin on the left here. And then on the right, we have the image of the 347 gene panel with more than 67 million counts of transcript assessed. And then by overlaying these two data sets, we again get additional information, not just about where genes are being expressed, but also where protein is being produced, which in a lot of different developmental and disease state studies is very important, not only to see where genes are being expressed, but also the end result in protein distribution and expression. And as I mentioned before, we support not only cell culture and fresh and fixed frozen samples, but also importantly, this FFPE preparation, which is very relevant clinically as a lot of clinical samples are preserved this way. We actually have a lot of publicly released data available on our website, which includes a lot of what I'm showing here. This is the using a 244-plex gene panel with these different tissue types. So you can see each pseudo color here represents a different gene overlaid on each of these images here. So we have ovarian cancer, lymph node, tonsil, lung cancer, and kidney. And I do want to point out this human skin here because this is a very difficult tissue type to dissociate. So it is very hard to assess in other sequencing modalities. But we have the ability and the support with our Merscope and our Merfish protocol to support this application. And of course, you can see here, successfully describe it with this panel. And once more to show the utility of the flexibility of the machine and the large imaging area and the ability to do different tissue types and preparations. We have a microarray of FFP human melanoma, 106 different cores over the imageable area of our slide using a 500 gene panel. And you can just extrapolate from there the different kinds of experimental design that you can use here using this large imaging area and the large gene panel to describe different samples on the very same slide. And the panels that we offer, I've already mentioned, are custom gene panels that are designed using our gene panel portal. And again, I wanted to reiterate that even though um, the support that the automatic workflow for the panel portal right now is for human and mouse, we support the design of panels for any species that can be associated with a well-annotated transcriptome. We can support panel design for that right now, up to 500 genes, with a 1,000 gene panel coming very, very soon. In addition to this, we also have pre-designed panels. So if a researcher is not sure how to go about designing their custom panel, or they just want some initial support on or driving a pilot study, we have pre-designed panels here. So on the left is the pan-cancer pathways panel for human tissue, and on the right we have the pan-neuro panel for mouse tissue. And we can provide gene lists for that upon request, and of course provide, again, support for custom gene panel design as well. And another thing we want to always highlight is that this technology, even though it is cutting edge and very new and it is highly utilized in the field already and rigorously validated. And we like to point that out because we have over, I think, last I checked, 112 peer-reviewed publications and preprints published on this technology and our platform in very high-profile, high-impact journals, as well as the institutions that are already using Merfish are very high-profile and impactful institutions that produce extremely rigorously validated studies. So this is all just to say that even though this is a new technology, our original science publication for Merfish was published in 2015. This has already been widely demonstrated for lots of different applications and many different tissue types by some very effective researchers. And getting more into the applications themselves here and some example publications that I was just referencing. We have a lot of really great impactful publications in neuroscience. So we've already talked about the cell atlasing in the hypothalamus, taking this a step further. Recently, there were 2 preprints released from the Allen Institute in Harvard detailing the whole mouse brain atlas. So these are really interesting, high throughput publications that these researchers were able to achieve by taking sections of the mouse brain, I believe every 200 microns, running them on our platform, and then they were able to register these samples and construct an entire whole brain atlas, which is very, very impressive. In addition to this, we have functional studies as well, in addition to the atlasing, and we will get into more detail about this agent study on the next panel or slide. But one thing I also wanted to mention was this memory engram study, which is also very interesting. This is from the Sudoff and Quake groups at Stanford. And they were able to create a spatial map of memory engram cells in the mouse amygdala and essentially identify a neuronal subpopulation, which was essential to long-term memory trace. So this is really unique work, and also it revealed the interaction with associated support cells and the consolidation of this memory. So really cutting edge research, and as a neuroscientist by training, I know that creating a physical map and description of memory or memory traces is something that neuroscience has been working towards a long time. So this is very exciting work. In addition to this, we also have lots of studies that describe disease mechanisms. So on the top right here, we have a study that describes different brain inflammation and infection pathways in the mouse brain. And on the bottom right here, I wanted to highlight this poster that was presented at SFN from us and collaborators at Columbia University. This is a study on presentation of Alzheimer's disease and pathology of Alzheimer's disease, which utilizes not only our gene panel, but also utilizes, again, you can see in the bottom row of these panels here, our protein co-detection. So again, this is another example of why being able to examine gene expression and protein expression and how those differ and align is very essential to lots of forms of research, but especially pathology of different disease. And as I said before, this is the mouse brain aging study that was examined using our platform. Essentially, what was done is over the course of the study, young, juvenile, and old animals were assessed in the prefrontal cortex and striatum, two nuclei of the brain that are known to be very affected by normal aging. And these researchers were examining the response in these animals to aging, normal aging, and immune challenge, and how both of those different presentations were either similar or different. And they were able to find spatially dependent cell state changes in non-neuronal cells, which is very important. This association of support cells and immune cells all working together in conjunction and how that interacts with aging and infection is a burgeoning area of neuroscience research. Um, and they found similarities between LPS challenge-induced brain inflammation and aging, which were very, very interesting. So if you're interested in this publication and learning more, please let us know, and we will gladly send you a copy of this publication, or you're able to find it by looking at our publication database on our website, which is sorted by category and searchable easily. So we don't just have a lot of high profile, interesting publications available for neurology and neuroscience research, but also many applications in oncology as well. So once more, we'll get more into the left-hand panel in the next slide. I have a whole slide breaking this down. But essentially, Merscope is used to study cell distribution and interrogate gene expression pathway in lung cancer in that study. And in the middle panel, this is a drug method of action study for a phase 2 clinical trial for hepatocellular carcinoma using a PD-1 blockade. So just looking at the different expression of genes downstream from this effector and looking at the response of different dendritic cells and T cells in response to this blockade. And this is important because, again, this is the a relationship between inflammation and immune-driven cancer, and being able to examine that whole pathway using our large panel size is really essential here. And then finally, again, relating to that tumor microenvironment and immune profiling, this is another study that was profiling 350 genes in colon and ovarian cancer and identifying the association of dendritic cells and activated T cells to, again, characterize a pathway in a tertiary lymphoid structure region. So this is an interesting piece of research, and I wanted to highlight it because this is part of a larger effort to generate a spatial tool called the Spatial Expression Explorer, which is a comprehensive analysis software and web-based application. So this Merfish data was used to demonstrate the utility of this tool and demonstrate the colocalization score method that they're developing with this tool. This is a 484 gene panel with patient lung cancer, and they were looking at the proximity of T cells to accompanied by inflammation and cell proliferation pathways. And essentially what they found was upregulation of kinases, RTK and MAPK, that they had not characterized before. And they were able to do this not only with the tool that they're describing in this paper, but specifically because of what the Merfish and Merscope technology enabled. So hopefully we have been able to describe all the advantages of this technology effectively, including the multiplexing, to be able to describe hundreds or soon to be thousands of genes using a custom gene panel and protein code detection on your sample over a large imaging area of intact tissue. This is enabled by the high resolution of our optics, so 100 nanometer resolution, which drives our ability to detect single transcripts and lends itself to our sensitivity and specificity for this direct RNA quantification. And in addition to this, it's a very flexible platform, which I think is very important for a lot of collaborative research that goes into these cutting edge publications. So you're able to be supported with any tissue preservation type, so cell culture, fresh frozen, fixed frozen, and FFPE. In addition to, you're not limited by your species of choice here or tissue of choice. We have protocols and knowledge for a lot of different tissue types and species types, and we have the support to be able to design panels for all of those as well. And if you want any more information, I mentioned our website earlier. It is a excellent resource. So not only are you able to explore more detail about publications, again, we have a searchable publication database, which is also sorted by research interest and application. We also have there are publicly released data sets that I mentioned a little bit earlier. So I mentioned our FFPE data set, which is our FFPE human data set. In addition to that, we also have a mouse brain data set and mouse liver data set. All of these are fully explorable on a web-based visualizer, which is on each of these releases web pages, and you're able to download the data as well to explore it on your own. If you need any more information about anything you've heard about today, please feel free to either contact my colleague Jose. His email is right there. He is the regional account manager for the Mid-Atlantic, as well as myself. Again, I'm Claire. I'm the field application scientist supporting this area. Or you may reach out to info@visgen.com. And I wanted to plug our social media as well as the last thing here, because we have a lot of exciting updates that we release regularly on our LinkedIn and Twitter. So please give us a follow. And I don't know if it's called Twitter anymore, X, I guess. But we regularly interact with researchers that are publishing these cutting edge publications, and we like to highlight them and also give out any information we can about any conferences we are attending, so you can speak with us in person about the platform if you wish. So with that, I think we have about 15 minutes left for discussion, so I will
Hi,
take a
Claire.
look.
Can you hear me?
Mm-hmm.
So there are a few questions on the chat window.
All right.
So the first one is around segmentation. So the question is the segmentation done on membrane staining images or reads?
That's a really great question. So we have two options for segmentation built into our software. So one is watershed segmentation, which is seated on DAPI stain, which is incorporated into all of our sample preparation. And the second is a cell pose driven algorithm. So this is an AI-based algorithm, which again, like your question asked, is based on membrane stain. So we support cell membrane stain. We have a kit for it that we can provide users to do this kind of segmentation. This segmentation is done in line with the processing of the images at the end of your run, and you as the user have the control over which algorithm is selected for your data.
And then the next question is around data handling. Please expand on data handling visualization, how or if Visgen can work with third-party and open data processing visualization platforms.
Great question. So the data that you get from each run, as I mentioned, is not only the VZG file format, which is directly importable into our visualizer, very convenient because it contains all of the summary information from the run, but you also get all of the raw data. So you get CSV files for cell by gene, cell coordinates, and transcript list, in addition to all of the raw images, the stitched raw images, and a parquet file with all of the cell segmentation as well. The data is easily transferable from either the instrument PC or the analysis PC. We have the ability to, you can transfer it off of your interconnection. We have a lot of people that have their machines networked to their internal servers, and they can do data transfer that way. There's also a USB-C connection at the front of the machine where you can manually offload data. And of course, because you get all this raw data, it is compatible with any downstream or third-party software that you would normally use for sequencing or quantification, gene quantification. We do offer support for VisGen post-processing tool. So this is a command line tool that we recently released that has the ability to resegment your data, so not using cell pose or not using watershed or using another cell membrane stain that you can incorporate with the protein code detection. This is, again, all open and not limited by any proprietary software that we have, although we do offer that wonderful summary file, the VZG file that you can examine immediately. And in terms of data handling, like I said, the capacity for our machines is really large, so it's 15 terabytes of storage available. But again, it's very easy to offload your data sets in a shared use environment.
Okay, and the next question is, how many probes per genes do we use?
I think that might be maybe proprietary information, but I believe it's a couple dozen to several dozen probes per gene. So that builds into the error robustness. So the barcode that you're seeing by the end of the run, which is used to decode the spot detection that is generated by the readout probes, is anywhere from 18 to 27 bits long, depending on the size of your panel. And that is generated again by attaching these readout probes to any of those binding sites on the encoding probe. So you can imagine how many probes would be per gene, and that lends itself to the error robustness. And again, the error robustness is also supported by blanks that are built in to your code book as well. So that is all to say that error robustness is built in with the number of probes per gene that we use. I believe, like I said, it's anywhere from a couple dozen to several dozen per gene. And that's where you get the tens of thousands of probes per panel.
Thank you. The next question is around panels. Do the custom panels come with from a pre-designed pool of options, or can one design entirely unique probes?
So the custom panels are whatever you can provide that is supported by an annotated genome. So if you can provide the ensemble ID or the sequence yourself and an abundance measurement, we can include that in a panel.
Okay, and there was a question about data. So there was a link shared by one of our colleagues that points you to our website where you can actually see the data that we are sharing through the website. There's another question coming in, I believe.
What is copy number per gene? And yes, the answer is the platform does detect copy numbers of individual transcripts. So when you look at your end data, let's say in the visualizer, you can actually hover over these individual spots. They will include the gene identification. So it'll say, GABBR1 when you hover over an individual spot. Copy number per gene just means the total amount of copies per gene that is in your panel, which again, we're directly quantifying with this barcoding system. I can see another question being asked here.
I think you probably answered the last two. Just one more coming.
And if there's anything that anyone thinks of after, let me just go back one slide. Make sure that this stays up. So if you have any questions that you think of later, please feel free to reach out to myself or Jose, and we will answer them as soon as possible.